Hello, NEET aspirants. Welcome to NEERAJA'S Biology Academy. You are with Akula Neeraja Prakash. In previous videos, I have mentioned you the importance of solving of previous year NEET papers. Since 2019, approximately 60% of the questions are sparked from the previous year papers. Now, I am uploading NEET 2021 question paper, linking with that of previous year NEET papers. Chances are there for us to get the questions right from this paper. Now I am uploading the questions related to the botany subject. Let us come to the first question. Which of the following statements is not correct? We observe this question appeared in all these years, and then so answer pyramid is related to the ecological pyramids. Pyramid of number in grassland is upright. Pyramid of biomass, so ANC is generally inverted. Pyramid of biomass in generally is upright. And this question appeared in AIMS and also in 2017 and 16. Pyramid of energy is always upright. So which one is incorrect here? The third option. Chances are there for us to get the question related to the diagram based. And then if we observe this in pyramid of energy, transfer of energy, so 1% of the solar radiant energy in case of green plants is converted into NPP. Only 10% of the energy is right from one trophic level is transferred to the successive trophic levels. Chances are there for us to get the limitations related to the ecological pyramids. If we come to this question, which of the following plants is monoecious? So the plants are considered as monoecious if both male and female flowers are present in the same plant. If both male and female reproductive organs are present, structures are present in the same flower, then it is known as a bisexual flower. If the male and female flowers are present on the separate plant, then it is considered as a dioecious plant. So here monoecious. So here in case of Cycas sarcinalis, Carica papaya, Cara, and then Marcantium, where Cara is a plant which is monoecious. You can come across in this diagram nucleus and globule. Nucleus is considered as female reproductive structure and globule is considered as male reproductive structures. The first stable product of carbon dioxide fixation in the sorghum is, so the sorghum, it exhibits a C4 cycle. And then phosphoglyceric acid is the first stable compound, a three-carbon compound associated with the C3 cycle. As a result of glycolysis, the end product that is produced is pyruvic acid that enters into the Krebs cycle. Then oxaloacetic acid is the first stable compound. It's a 4-carbon compound which is formed in the C4 plants. So how many times every year we are getting the question right from this specific area? Chances are there for us in this year to get the question right from photorespiration or C2 cycle. And then this particular photorespiration, it was coined by Krokov. The current PO or the scientists, they observed in case of tobacco leaves. When the temperature, when the intensity of light and oxygen is more, in such conditions, instead of photosynthesis, the phenomenon photorespiration will occur. In this particular condition, Rubisco enzyme, instead of functioning as a carboxylase, it functions as an oxygenase. For the purpose of photorespiration, chloroplast, peroxisomes, and then mitochondria are involved. And then the energy liberated is zero. It is a wasteful reaction. Diadelphous stamens are found in case of Chinaros monoadelphous condition is observed in case of diadelphous condition is observed in case of Fabaceae members. Polyadelphous condition is observed in case of citrus plants. And syngenaceous condition is observed in case of sunflower. And then St. Andrews condition is observed in the members of Cucurbitaceae members. We got a question in 2016 related to the polyadelphous. And then here in 2016, we got, they have given several examples and they have asked us to pick out which is a tetradynamous and didynamous. So tetradynamous is a condition where 6 stamens are present. It is observed in case of the members of Brassicaceae where outer stamens are short in nature and the inner stamens are elongate in nature. Whereas in case of a didynamous condition, it is observed in case of Salvia. It is not Salvinia. DNA strands on gel stained with ethylium bromide when viewed under UV radiation appears. So in related to bright orange red color bands were obtained. And then normally it's through the method gel electrophoresis we can separate the DNA fragments, and that method is known as southern blot technique. Whereas if RNA fragments are separated, then it is known as northern blot technique. In western blot technique, where proteins are separated. Once you check this diagram, show this particular area where lane 1 is associated with undigested fragments. Western blot technique is a method used to separate various different types of proteins. As the proteins are associated with different types of charges, it is very difficult to separate them through this electrophoresis method. So, in order to bring all the proteins or polypeptide chains to one charge, sodium dodecyl sulfate is used. In one of the previous years, we got this SDS in the options. Plant follow different paths in response to the environment or phases of the life to form different kinds of structures. And this ability is known as plasticity. The ability of one genotype to produce more than one phenotype is known as a plasticity. It may occur in the phase of their life or related to the environmental conditions. So here it is a one type of genotype, it produces more than one phenotype, whereas pleiotrophy is related to where the multiple traits by a single gene is known as a pleiotrophy. So this is a small difference associated with respect to the plasticity and the pleiotrophy. Pleiotrophy we will come across in case of sickle cell anemia and also phenylketonuria. Match the list 1 with that of list 2, protoplast fusion and then plant tissue culture, meristem culture and micropropagation. Somatic fusion is also known as a protoplast fusion, where two distinct species plants were collected and the protoplast it was separated, and then these cells are fused by using a polyethylene glycol, and then fusion of cells and then fusion of nucleus will occur, and then somatic cell hybridization occurs. So this is an example, tomato, where potato and tomato are present in the same plant. Related to the meristem culture, where in order to produce virus-free offsprings and multiple, where multiplication is very rapid related to the meristem culture. In case of our sponges, archaeocytes are totipotent cells, and then meristem culture is related to further produce virus-free plants. Micropropagation is associated with the production of soma clones. In the equation GPP minus O is equal to NPP. We got this question many a times. The total amount of organic matter that is produced by the green plants during the process of photosynthesis is known as GPP. And then the amount of energy that is required for respiration is represented here in the form of R. And then NPP is net primary productivity. Only 1% of the solar radiant energy is converted into NPP. Chances are there for us to get the problem right from this particular area. And then here, if we come to the options, retardation factor related to the chromatography, retardation factor we will come across. And retardation factor is equal to, where distance traveled by the substance by distance traveled by the solvent front. The production of gametes by the parents. Formation of zygotes in the F1 and F2 plants can be understood from the diagram. Called, which is known as a Punnett square. So this is a question we got in 2019 as such in Orissa paper. Which of the following is the incorrect statement? So in cell biology, every time each and every cell organelle is important. And then mature sieve tube elements that possess a conspicuous nucleus and then cytoplasmic organelles. No, sieve tube elements are not associated with the nucleus and also cytoplasmic components. They are dead cells associated with the phloem. And then microbodies are present in the plants and then animal cells. So microbodies are the structures which are associated with them. They are present in the peroxisomes. They contain hydrogen peroxide and they contain the enzymes that break down the hydrogen peroxide. The kidney and liver in case of vertebrate organisms and is involved in the oxidation of fats. Match the list 1 with that of list 2. Each and every paper, match the list 1 with that of list 2. So every year we are getting a question right from this particular area. In this year, we got a question. In 2021, we got the question. So 2 questions right from this particular area related to the secondary growth. Lenticels, these are responsible for exchange of gases. Related to the cork cambium, it is known as phloem. And the secondary cortex is associated with the phloem. And then cork is associated with the suberin deposition. So cork is known as phloem, cork cambium is known as phloem, and then secondary cortex is known as a phloem, together known as a peridome. Lenticels are seen in case of dicotyledons, which are mainly responsible for the exchange of gases. In 2020, we got a question related to the wood. Heartwood is the central part of the wood having more amount of extractives or secondary metabolites like resins, tans, or so gums, and then all these are the substances present in the heartwood. And in sapwood, it is peripheral wood. It is a peripheral wood. They dark in color, light in color. Chance are there for us to get this particular question. Physiologically inactive and physiologically active in nature. It is mite resistant. It is not mite resistant, and it is associated. So it is used for furniture making, whereas it is not used for the furniture making. The amount of nutrients such as carbon, nitrogen, phosphorus, and then calcium present in the soil at any given time is referred as, and it is known as standing state. Standing crop, the total amount of biomass that is present in the ecosystem at a given time, and then lowest standing crop is associated in case of oceans. Chances are there for us, so because we got this particular question. That is one thing. Emphasize on this particular ecological succession. And there's a primary succession and secondary succession. Primary succession, it starts right from the zero life area, and then whereas secondary succession, it is associated. So only after the already existing community, if it has undergone the perturbance, and then next, the term used for transfer of pollen grains from anthers of one plant to the stigma of different plants during the process of pollination. This is a very, very, very important area. So every year we are getting questions. In 2020, we got 2 questions right from this particular pollination area. Cleistogamy is related to the pollination within the closed flower. And then example, Pomelina, Volvus, oxalis, and Viola. And then chasmogamy is observed in case of the closed. Autogamy, where the transfer of pollen grain right from the same flower to the stigma. And then right from methanogamy, different flowers of a same plant, transfer of pollen grains if it occurs. Xenogamy represents the transfer of pollen grains right from the different plants of the same species. So you might have come across this particular, so autogamy and methanogamy are prevented in case of papaya because it is a dioecious plant. In case of maize, male flowers and female flowers are present on the same plant, and it prevents autogamy but not methanogamy. Which of the following stages of meiosis involves division of a centromere? In general, division of centromere, it occurs during anaphase stage. During meiosis 1, division of centromere does not occur. And then especially during anaphase 2. Anaphase 2, division of centromere is associated. In previous years, the attachment of spindle fibers to the kinetochore had become evident during metaphase stage. And then after meiosis, the resultant daughter cells have twice the quantity of DNA when compared with that of gamete. Anaphase promoting complex is a protein degrading machinery for the proper mitosis of the animal cell. If any defect is associated, then chromosomes they will not segregate, and it leads to non-disjunction. Which of the following is the correct sequence related to the steps in case of polymerase chain reaction? Stages: denaturation, annealing, extension, or the stages associated with the polymerase chain reaction. When gene targeting involving gene amplification is attempted in an individual tissue to treat the disease, it is known as gene therapy. First time gene therapy was given in year 1990. A girl who is suffering from SCID due to the non-functional activity of ADA gene. Her name is Ashanti De Silva. So ethical issue, GEAC, where organization responsible for assessing safety introducing the genetically modified organisms for public issue. It is known as a toxicity or safety testing. It is a procedure where transgenic animals are made to carry a particular type of gene which is sensitive to the toxic substances, and thereby they can compare the effects when compared with the top non-transgenic animals. And then so biowar, weapons or anthrax and then botulism. We got this question in 2019. Match the list 1 with that of list 2. Every year we are getting questions right from the cell biology in the form of statements or in the form of match the following. If you observe this question, you will shock. So this is the question as such. It is picked right from 2015. So here you can see thylakoids, cristae, and then cisternae. Here also cristae, thylakoids, and then cisternae all are same. The factor that leads to founder's effect in a population is genetic drift. So I have picked the content related to the natural selection, which is scattered all over the NCERT book here and there. When we describe the story of life on the earth, so here it is evolution, it is a consequence of natural selection. So natural selection, it leads to stabilizing selection, directional selection, and disruptive selection. Stabilizing selection, where more individuals acquire the mean value in the form of some statements, chances are there to get derived from this particular area. You can go through disruption and also directional selections. When the centromere is situated in the middle of two equal arms of the chromosomes, the chromosome is referred to as, is known as a metacentric type of chromosome. So you can come across this particular different types. It is an acrocentric type of chromosome, and it is submetacentric, and it is metacentric, telocentric, and then this is the chromosome which is having satellite region. The shape of the chromosomes can be visualized during the Anna phase. And then this question we got in 2019, the small arm of the chromosome is denoted by P arm, whereas the large arm is denoted by Q arm. Germe are present in case of liverworts or in case of Mercantia. In case of mosses, by the process of budding, small filaments are formed right from the protonema. That is asexual reproducing structure. Gemmules are the structures, asexually reproducing structures seen in case of sponges, especially in case of freshwater sponges. During the purification process of recombinant DNA technology, chilled ethanol precipitates out. So here DNA. You have to focus on the structure related to the histone proteins. So chances are there for us to get some questions right from the histones. So how many types of histone proteins are there? So how many types means 5 types. And then what is the structure associated with the nucleosome? Nucleosome, how many turns of DNA are present in case of nucleosome? And then what is the, how many base pairs are associated with each nucleosome? Like that, several chances are there for us to get the question related to the histone. And these are histones or the basic proteins which are associated with the lysine and the arginine. Match list 1 with list 2. So here this is related to the ascent of sap. Ascent of sap, where upward movement of water and minerals against the gravitational force, and also according to the water potential gradient. And then here different types of physical forces that are responsible for entry of water right from the soil to the plants. Cohesion, cohesive force, adhesion, and also surface tension. Water molecules are held together by, and they form unbroken form, and they form the column. So water column, which is known as a cohesion. Water is held to the polar surface, then that condition is known as adhesion, especially to the xylem vessels. And then water held more in the liquid state than in the gaseous phase, which is known as a surface tension. And then guttation is a phenomenon where the loss of water, it occurs in the form of droplets. Hydrodes are the structures which are responsible for guttation. The site of perception of light in plants during photoperiodism is the leaf. Duration of the available light into the plants is known as a photoperiod. The expression of the plant in the form of flowering due to the photoperiod is known as photoperiodism. And then normally the requirement of light by the plant is known as a critical duration. Photoperiod in case of a particular area so is the same for all the plants, but critical duration it differs in case of different types of plants. And then a particular hypothetical hormone which is required for this flowering in case of the plants is florigen. The plants are differentiated into short day plants and then long day plants and then day neutral plants. Example for short day plants, xanthium, strawberry, and also rice. Chance are there for us to get the question related to the vernalization. It is an artificial exposure of the plants to low temperature to stimulate the flowering and then to enhance the seed production. And it prevents precocious reproductive development. So late and then reproductive development enables the plant to have sufficient time to reach the maturity. The plant hormone used to destroy the weed. So the weed in the fields. 2,4. So different types of phytohormones are present where there are auxins, gibberellins, cytokinins, and ethylene, and abscisic acid. So indole compounds where there are auxins, different types of auxins are present. They may be either natural auxins or synthetic auxins. Natural auxins are so IAA and IBA are natural auxins, whereas synthetic auxins are so where NAA and 2,4-D are synthetic auxins. 2,4-dichlorophenoxyacetic acid is common. So synthetic herbicide that is used to control the weeds, especially which are having broad leaves. Which of the following algae produces carrageen? So carrageen is produced by the red algae. Blue-green algae, which is a cyanobacteria. And then green algae, chlorophyll member. Brown algae, so phaeophycean member. Red algae, it is a rhodophycean member. So here we have to focus on what are different types of chlorophyll pigments or stored substances, stored reserve food substance, where it is starch, mannitol, laminidin starch, and then fluorine starch is associated. What are the compounds that are present in case of cell wall? Very, very, very important area. Go through this particular table. Complete the flow chart on the central dogma. Central dogma of the protein synthesis is DNA. It gives rise to mRNA, and that mRNA gives rise to mRNA gives rise to the protein. So here DNA is produced as a result. This process is known as replication, and then this is known as transcription, and this process is known as translation. As a result, a protein is produced. For the replication of DNA, different types of enzymes are required. Helicase, topoisomerase, SSB protein, DNA polymerase 1, 2, 3. All these are various types of proteins associated with the replication of DNA. And then transcription in case of prokaryotes, transcription in case of eukaryotes. RNase is the enzyme. DNA dependent RNA polymerase enzyme is essential for replication, transcription in case of prokaryotes. And then mRNA is synthesized where for this, where sigma factor and also rho factors are associated later to the transcription in case of prokaryotes. In case of transcription of eukaryotes, 3 different types of polymerases, RNA polymerases are associated. RNA polymerase 1 is involved in the synthesis of rRNA and also 28S and then 18S and 5.8S. So rRNAs are produced, whereas RNA polymerase 3 is involved in the transcription of tRNA and also 5S RNA. And then so RNA polymerase 2 is responsible for production of heterogeneous nuclear RNA or hnRNA. Which of the following are not secondary metabolites in case of plants? Secondary metabolites in case of secondary metabolites where quinine, which is extracted from Cinchona officinalis, which is used as an anti-malarial drug, nicotine, which is produced right from the tobacco plant, which is used as an insecticide, morphine, right from Papaver somniferum, it is used as a painkiller, and the reserpine, which is present in the lobule, which is extracted from Rauwolfia ometeria, which is normally which is an anti-hypertensive drug, whereas reserpine is also used as a medicine for the snakebite. Caffeine, which is extracted right from Cochia arabica, which is used as it is a stimulant and the central nervous system. It acts on the central nervous system. And then pyrethroids, which is extracted right from chrysanthemum, and it is used as an insecticide. Along with this, trichine, cardiac glycosidases, all these are also secondary metabolites which we are having this in organisms and population topic. In spite of interspecific competition in nature, which mechanism of competing species might have evolved for their survival? Related to predation, one organism will survive, another organism will not. Whereas resource partitioning, and then MacArthur is a scientist. He gave the concept related to the resource partitioning. Competitive release, where competitively superior organisms will survive, whereas another organism cannot survive. Gauss principle explains a competitive release. Which of the following is not application of a polymerase chain reaction? So detection of gene and then molecular diagnosis and then gene amplifications are associated. Purification of isolated protein is related to the downstream processing effect. Following applications of PCR reaction, so where amplification of the gene fragment, modification of the DNA fragment, sensitive detection of the pathogenic microorganisms, and DNA analysis of the archaeological specimens. And then it also used to detect the specific type of diseases related to cystic fibrosis and also Parkinson's disease. It is also used to detect the gonorrhea, chlamydia, which are sexually transmitted diseases. Match the list 1 with that of list 2. The cells with active cell division capacity is meristematic tissue. Tissues having all cells similar in the structure and their function. They are known as a simple tissue, parenchyma, collenchyma, and the sclerenchyma. And these parenchymatous cells, they have the ability to undergo the process of the differentiation and is transformed into the issues cambial activity. Tissues having different types of cells are vascular tissues. Xylem and then phloem are considered as vascular tissues, where xylem is associated with the trachea and then xylem fibers and then xylem parenchyma. In case of phloem, where sieve tube and then companion cell, phloem parenchyma and then phloem fibers are present. Xylem parenchyma cells are live cells related to the phloem. So phloem fibers are the dead cells. Dead cells with the thickened walls and a narrow lumen is the sclerites. A typical angiosperm embryo sac at maturity is associated with 8 nucleate and 7 celled stage. Related to the development of embryo sac, megasperm undergoes 3 successive karyokinetic divisions takes place. As a result, 8 nuclei are formed. After third karyokinetic division, where cytokinesis occurs, consequently, it gives rise to 7 cells. The structure related to the embryo sac, which consists of chenacil end and micropylar end. 3 antipodals, 3 cells of the egg apparatus, and then 1 at the central cell. Totally 7 cells and 8 nuclei are present. Which of the following algae contains mannitol as a reserved substance? So where brown algae or the organisms which possess mannitol or laminarian starch as the reserved substance. Here, Ectocarpus is an organism which consists of mannitol as a reserved substance. Related to the Elothrix, Elothrix is a filamentous unbranched algae and it shows vegetative propagation by means of fragmentation. Aplanospores are responsible for asexual reproduction and also zoospores are related to that of sexual reproduction. Volvox is a colonial green alga, popularly known as rolling alga or rolling ball, and the colony is known as Cenobium. It contains nearly 500 to 60,000 flagellated interconnected cells in that particular ball-like structure. Asexual reproduction takes place by the formation of the daughter cells and which is present within the parental body, whereas sexual reproduction is oogamous type. Genera like Selaginella and Salvinia, they are heterosporous in nature. In case of pteridophytes, all the plants are homosporous except Selaginella and also Salvinia. Here the Silaginella plant it shows dichotomous branching. The area where branching is associated, right from that particular area, the roots are formed. Once we got a question related to Salvinia in one of the Ames paper. Salvinia is associated with the three different types of leaves. Roots are absent in case of Salvinia. And then first type of leaves are broad in nature and are useful for photosynthesis. Second type of leaves are meant for floating. And third type of leaves are highly dissected and they compensate the absence of roots. And if you observe the equisetum, it is associated with rhizomatous stem. Here it is known as a rhizome. Rhizome is present in case of equisetum. Mutations in plants can be induced by zeatin, kinetin. These are cytokinins which are not associated with the mutations. It is possible to induce mutations artificially either by subjecting the plants to the chemicals or by the radiations. In case of mung resistance to yellow mosaic virus and then powdery mildew were induced by mutations. The resistance to yellow mosaic virus in case of bindi, where Abelmoschus esculentus is developed by mutations and now it is known as a Parbhani Kranti. Amensalism can be represented by. So these signs you have to follow. And amensalism, one organism is neither benefited nor harmed. One organism is adversely affected. Antibiotics are example for amensalism. And then what is the role of RNA polymerase 3 in the process of transcription in case of eukaryotes? It is involved in the transcription of tRNA, 5S RNA, and also snRNA. Select the correct pair. Loose parenchymatous cells rupturing right from the epidermis form the lens-shaped opening and also opening of the bark, which is known as a spongy parenchyma. So these cells are complementary cells. Large cells, large colorless empty cells in the epidermis of the grass leaves is known as a subsidiary cells. They are known as bulliform cells. In dicot leaves, vascular bundles are surrounded by large thickened walls and then which is known as a conjunctive tissue. Conjunctive tissue is present in case of roots which are present between xylem and phloem. Cells of the medullary rays, they form the part of the cambium. Yes, it is a correct statement. Plasmid PBR322 has PST1 restriction enzyme within the gene also for ampicillin resistance. That confers ampicillin resistance if the enzyme is used for inserting a gene for beta galactoside. Production and the recombinant plasmid is inserted on E. coli strain. So then what will happen? It is a hypothetical question. So what will happen? It will not able to confer ampicillin resistance to the host cell. So if we observe the structure of PBR322, it is related to this particular area which is associated with the ampicillin resistant medium and also tetracycline resistant areas. Ampicillin resistant area is associated with two different types of restriction sites where Proteus vulgaris 1 and also Providentia stewarti. Whereas tetracycline resistant area is associated with the BAM H1, Bacillus amino, liquefations, and also CAGCTG and it cuts the blunt ends. DNA fingerprinting involves the identification of differences in the some specific regions in the DNA sequence called, which is known as repetitive sequences. Repetitive sequences which are not expressed in an exponential growth equation, NT, N0, ERT, it represents a base of natural logarithm related to the logistic growth, where dn by dt is equal to r in k minus n by k. Once in 2016, we got a question related to this particular area. If it is, chances are there for us. So here, arithmetic growth in case of plants and also exponential growth in case of plants. The arithmetic growth in case of plants is mathematically represented as LTA0 plus RT. And then, so here, where LT is related to the length of the time, and then L0 at the zero time, or is the growth rate elongation per unit time. Related to exponential growth, W1 is equal to W0 ERT. Chances are there for us to get right from the particular diagram, arithmetic growth and also geometric growth. Which of the following statements is incorrect? Process of photosynthesis, it includes light-dependent and light-independent reactions. Light-dependent reaction is associated with the cyclic and then non-cyclic photophosphorylation. Related to the non-cyclic photophosphorylation, the reaction center is 680. When the light energy, which is associated with the 680 nanometers, so energy and immediately photolysis of water will occur. As a result, electrons are released right from the reaction center, and those electrons are accepted by primary electron acceptors, and these electrons are passed to plastoquinone and then cytochrome B6 complex acceptor and also plastocyanin. And then finally, right from plastocyanin, it reaches the photosystem 1. Electrons right from the primary electron acceptors of the photosystem 1. And it is transferred to the plastoquinone and also to the flavoprotein. Finally, NADPH is formed. Whereas the ATP energy is also liberated here at this particular stage. Cyclic photophosphorylation includes PS1 and PS2. No. Both ATP and NADPH are synthesized during non-cyclic photophosphorylation. Yes. And then stroma lamella PS1, it lacks the NADP reductase. Which of the following statements is correct? So here, fusion of protoplasm. So between two motile, non-motile gametes is known as plasmogamy. Match column 1 with column 2. This is also another important area to focus each and every microorganism. If we come to the metabolism of nitrogen in case of green plants, the first step is the fixation of nitrogen. It may occur either by biological method or by industrial nitrogen fixation. To biological nitrogen fixation, nitrogen is converted into ammonia. In 2020, we got a question right from this particular area. And the enzyme which is required here is a nitrogenase. So different types of microorganisms are involved in the nitrogen fixation. These organisms may be symbiotic organisms or non-symbiotic organisms. Rhizobium is associated with the legumes. Frankia is associated with the non-legumes. Anabina is associated with the Anabina. It's a blue-green algae which is associated with some non-legumes, and also Astrobacter, which is a free-living organism. All these organisms are involved in the fixation of nitrogen. And then nitrification, where this nitrates, where ammonia is converted into the nitrites in the presence of nitrosomonas and nitrococcus. Nitrites are converted into nitrates in the presence of nitrocystis and also nitrobacter. Nitrates which are formed are taken up by the plants, but again they have to convert into ammonia, for which nitrate reductase enzyme is required, and then in the presence of molybdenum, this enzyme is activated. So nitrite is converted into ammonia, and then produced ammonia is involved in the synthesis of amino acids. So synthesis of amino acids takes place in the leaves. Now you can pick out the answer for this particular question. And the next, which of the following statements is incorrect? It is another important area. Oxidation reduction reactions produce proton gradient in the respiration phase. During aerobic respiration, role of oxygen is limited to the terminal state. And then electron transport system. So transports one molecule of NADH gives rise to two molecules of ATP. So one molecule of NADH gives rise to three molecules of ATP, and then FADH gives rise to two molecules of ATP. And then ATP is synthesized through the complex 5. Yes, if we come to the electron transport system, it is associated with totally 5 complexes. And then 3 complexes are pakka involved in the transport of electrons and then protons. And then one complex is associated with the synthesis of ATP. If we come to the first complex, so which is known as is a NADH dehydrogenase. It is involved in the transport of protons, a translocation of protons, and also transfer of electrons. So here it is NADH dehydrogenase enzyme. And then the released electrons are taken up by the electron carriers by FMN, and then it is conveyed to iron sulfur complex, and then are conveyed to ubiquinone, where it is converted into ubiquinol. And then the next complex is complex 3, cytochrome C reductase complex, where the electrons which are taken up by cytochrome B and then so C are conveyed to cytochrome C, which is an electron carrier present outside the membrane. And then it is a mobile carrier, where this complex form is receiving the electrons right from the cytochrome C. So it is considered as cytochrome C as an oxidase enzyme. Complex 2 is involved in the liberation of electrons. And then these electrons are donated to the ubiquinone, where it is transformed into ubiquinol. And then the complex 3. Complex 1 and complex 3 are involved in the translocation of 4 protons each, whereas complex 4 is involved in the translocation of only 2 protons. Complex 5 is involved in the synthesis of ATP. In some members, which are the following pairs of the families, pollen grains retain the viability for the some months after their release. So this is a question, direct NCERT question where so Rosaceae, Leguminosae and Solanaceae members they can be. And then it is possible to store the pollen grains the large amount for the longer period of time in the liquid nitrogen by the method known as a cryopreservation at 196 degrees centigrade at so where the pollen grains are stored. Identify the correct statement. So here split genes arrangement in the characteristic feature is observed in case of prokaryotes. No. Split genes are seen in case of eukaryotic organisms. Capping under methyl guanosine triphosphate is added to 3' end of the HNRNA. It is added at 5' end of the HNRNA. And then RNA polymerase binds with the Rho factor to to terminate. Sigma factor is attached to the RNA polymerase and plays an important role in the process of elongation. In case of prokaryotes, Rho factor, it binds to the polymerase, RNA polymerase, and favors the process of termination. Match the column 1 with that of column 2. Same type of question we got in 2020. The form of match the following. So Liliaceae members can be detected by having 3+3. Solanaceae members, united, they are gamopetalae members. Brassicaceae members, where by observing the stamens we can detect the Brassicaceae members. So match list 1 with that of list 2. Right from this particular area, we used to get the bonds related to the biomolecules. We used to get the bonds, different types of bonds. And then the structure of DNA. What is the length of the DNA? Problem chances are there for us to get the problems right from this particular area. And then related to the enzymes, holoenzyme, coenzyme, cofactors, all these are important related to the enzymatic part related to this particular chapter. What are different types of bonds that are found? Proteins are associated with the peptide bonds. Whereas based on different types of structures, again, different types of bonds are associated. But and then unsaturated fatty acids are associated with double bonds. And then nucleic acids are associated with the phosphodiester bonds. And a polysaccharide is associated with the glycosidic bonds. Last year, we got a question related to the bonds that are present in maltose, sucrose, and also in case of lactose. In case of maltose, alpha 1,4 glycosidic bond is present. In case of lactose, beta 1,4 glycosidic bond is present. In case of sucrose, alpha 1 and beta 2 bonds are present. In case of glycogen, so normally a straight chain is associated with alpha 1,4 glycosidic bond, whereas in the branched regions, alpha 1,6 glycosidic bonds are present. In case of starch, alpha 1,4 glycosidic bond is present. In case of cellulose, beta 1,4 glycosidic bond is present. Match list 1 with that of list 2. It is another important area. Every year we are getting 2 questions right from this particular area. Cell cycle is associated with the interphase and then mitotic phase or division phase. Interphase is divided into G1 phase, S phase, and G2 phase. G1 phase is the actively preparatory phase, and it is present between interval between mitosis and initiation of DNA replication. Sometimes the cells, some of the non-dividing cells, they exit the G1 phase and they remain at the stage of inactive phase, which is known as G0 phase. During S phase, replication of DNA takes place. As a result, 2C content is doubled and reaches to 4C, but division of chromosomes does not occur. And if you come to the G2 phase is the gap 2 phase where tubulin protein is synthesized and then mitochondria, chloroplast, Golgi complex, all these are duplicated. Nowadays, it is possible to detect the mutated gene causing cancer by allowing radioactive probe to hybridize the complementary DNA in a clone cells. If a person is without any mutated genes, then the radioactive labeled probes will go and bind, and that can be expressed on the photographic film. If mutation has taken place, where the probes they cannot go and bind, and then they cannot be represented on the photographic film. By observing this, you might have come across the importance of solving of this current 2021 paper, 2019, 18 papers. Already this concept I have uploaded 35 days back in one of my videos. All the very best. Meet you in the upcoming videos. Thank you.